Ya, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Pada kesempatan kali ini saya akan membahas tentang Bilingual Teaching RNA Virus, materi kelas 10 SMA dengan mata kuliah Metodologi Pengajaran dan dosen pengampu Ibu Doktoranda Nurmasari Sartono M.Biomed. Oke, selamat menyaksikan.
Okay, assalamualaikum everyone.
Waalaikumsalam.
Good morning, good afternoon. How are you today?
I'm fine, thank you. And you?
Oh, I'm very good. Thank you very much for your attention. Okay, how about your feeling during the fasting in the Ramadan? I hope you will feel enjoy and happy. And now I will make a presence of you first. Kamal Akhtar Adawi.
Yes.
Okay. Because what we are going to discuss in the meeting is very popular matter. Today we are going to talk about the virus classification, but I'll focus only on RNA virus. So I will talk about what is RNA virus and how about the classification about RNA virus, and then how RNA virus can be injected in the host cell. Okay, so we go at the first. What is the RNA virus? RNA virus is a virus which has material genetics contain RNA, but they didn't have an DNA. Okay, you can see at the monitor. This is the past system of virus classification. You can see that the core classification is about RNA, such as retrovirus and HIV virus that will make an AIDS sick, and the second one is DNA virus, and then it will be more classified by the single strand or the double strand in the DNA or RNA. And then it will be classified by non-segmented and segmented virus. Okay, we'll next on the next paragraph. The virus also classified by the capsid. The capsid is the what can see the protector from virus from the immune cells in the human or in the animal body. And virus have membranes surrounding capsid, what I said, and animal virus such as HIV are frequently enveloped. Yeah, you can see in the picture capsomer of capsid, capsomer of glycoprotein, and in the tobacco mosaic virus, in the adenovirus, in the influenza virus, and in the bacteriophage or T4 virus. Okay, so this is the latest classification of the virus. Jadi ini klasifikasi tentang virus yang paling terbaru, which has been found by the Baltimore, which won the Nobel Prize in 1970. In 1970, the Baltimore classification said virus according to how the mRNA produced during the replicate cycle of the virus. And also, virus can contain double-strand DNA, single-strand DNA, or double-strand RNA, single-strand RNA, and by its polar charge, like a positive charge or negative charge. You can see in the next slide. Yeah, you will see in the pointer. The group 1 is the dsDNA virus, which can be positive or negative charge, and with also ssRNA virus, which is positive charge, and then SSR. Yeah, the DNA virus, RNA virus, and rapid transcription virus. Okay, we'll next in the next slide. RNA, not only the DNA as a genetic material by the virus, as I told the past week, but RNA can be genetic material in the virus. So, the broadest variety of RNA genome is found in viruses that infect animals. So, the RNA virus first is invented was injection the human body or the animal body, and then retrovirus is have a unique enzyme which can we say as a reverse transcriptase enzyme can make an RNA from the material genetics to be DNA in the host cell. And then HIV is the example of the retrovirus that can be caused an AIDS or acquired immunodeficiency syndrome. So your body will not be protected again. Di tubuh kita nggak mungkin akan nggak akan terlindungi lagi oleh sel imun gitu. Okay, the next we can discuss about the how RNA virus replication. First of all, the virus which is in the example of the image is HIV virus, and they have a very important thing in the HIV virus. This is reverse transcriptase enzyme. Okay, so first of all, the virus is attack the immune cell, which can the example of immune cell is the T clear cells. They came to the T clear cell. Okay, we'll discuss about how RNA virus can be injected and can be infected the host cell, which is a health cells. So the RNA virus will come to the host cell, and then they make a contact. Then the cells have a receptor. The receptor is a protein. We can call it CD4 and the CCR5. CD4 and CCR5 is fusion, makes the HIV virus and the cell virus fusion, and then the genetic materials which contain RNA reverse transcriptase enzyme, integrase enzyme, and protease enzyme enter to the cells. And yes, in the cytosol of the host cell, the RNA virus, the material genetic of RNA virus, which is RNA, is going to be a DNA by the reverse transcriptase enzyme. So we can see in this image, the first is RNA virus, and the RNA is make a DNA by the reverse transcriptase enzyme, and then it make a single strand of DNA. But the virus is make but the virus make a double strand of the DNA by the reverse transcriptase again, so they reverse again, and after that the HIV have the same DNA of the host cell. And yes, after the HIV have a DNA as a host cell, they can move easily to the nucleus. They are grabbed by the integrase enzyme and will carry out to the nuclear pore. Nuclear pore will carry out to the nucleus cell. In the nucleus cell, by the integrase enzyme, the DNA virus is connected with the chromosome of the host cell. Yes, you can see the provirus is injected in the nucleus cell, which is connected to the chromosomal DNA of the host cell. And after that, it will make a long life injection. Why? Because if the cell will make the protein, it will contain an RNA from the HIV virus. Okay, so the chromosomal DNA is make a messenger RNA. Messenger RNA is produced by the enzyme, which we can call it by polymerase enzyme. The polymerase enzyme will make the RNA virus, will make the messenger RNA, and the messenger RNA will go to the cytosol, and in the cytosol they will make a protein by the help of the ribosome. We will make the protein, and through the surface cell or the membrane or the cell membrane by the vesicle, which came from the upper Golgi apparatus, and then the other messenger RNA. So the HIV virus doesn't make one messenger RNA, but they make two messenger RNA. The other messenger RNA to make a protein which contain the material genetics such as enzyme and the double strand of RNA. And after that, the double strand of RNA is made into the new virus. The new virus is immature, but don't forget about the enzyme. They produce protease enzyme which can be make the immature HIV virus will make the it mature. Jadi tadinya belum matang, selnya dibuat matang dan siap untuk menginfeksi lagi dengan bantuan enzim protease. Okay, I think that's all. And okay, we go on the conclusion. In conclusion, don't forget there are several classification of virus, which is the past classification from DNA or RNA classification or by capsid classification, but the latest classification is made by is invented by the Baltimore in the 1970s. And RNA or DNA virus, RNA or DNA is a genetic or virus. So in the virus, there's only one DNA or RNA. And then the RNA virus has reverse transcriptase enzyme and the other enzymes such as protease, integrase, and others. And the example of RNA virus is HIV virus, coronavirus, and retrovirus, and etc. Okay, I think that's all for me. Anyone, do you have any question?
No.
Okay, thank you very much for your attention. I hope you will enjoy the material of this class, and I hope you will be more diligent to read a book about the virus. Okay, and the next meeting we will discuss about the bacteria, archaebacteria. So don't forget to look at your book and read the book, and see you in the next meeting. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.